Good morning, everyone, and welcome to the Citrus Research Board and UC IPM webinar on Asian citrus psyllid detection and IPM strategies. I'm Stephanie Pereira with the UC statewide IPM program. Peter Cosina is also here with me, and he'll be running the polls and troubleshooting any technical problems. We're also here with Joey Mayorkin from the Citrus Research Board, who will be introducing our speaker shortly. Please also note that this webinar is targeted to growers and agricultural pest management professionals. Master gardeners can certainly benefit from participating, but the pest management methods presented, especially the pesticides, are not to be followed without a clear understanding of their legal use in home environments. And now I'll hand the mic over to Joey and he'll introduce our speaker.
Thank you, Stephanie. Please welcome today's speaker, Dr. Monique Rivera, Assistant Cooperative and Extension Entomology Specialist at the University of California, Riverside. Prior to joining UC Riverside, Dr. Rivera worked in many different cropping systems along the East Coast, including postdoctoral studies at the University of Florida, where her research focused on vector-borne diseases such as Huanglongbing and laurel wilt. As an Assistant Cooperative Extension Specialist, Dr. Rivera's research focuses on the entomology issues growers find most important, with a focus on vector-borne diseases, Persea mites, among others, as well as pollination enhancement. Today, Dr. Rivera will discuss the current situation of the Asian citrus psyllid, its detection, and IPM management strategies in commercial orchards. Monique, please go ahead and share your screen to begin the presentation.
Okay, so getting started with today's talk, I am going to be talking to you about 4 different topics. Scouting strategies for Asian citrus psyllid, management of their populations once they're in the field, new technologies that my lab is studying, and then an overview of a newly funded scouting project. So getting started with scouting, let's first think about ACP movement and why do they move. They're not great flyers, but there are reasons for which they will decide to fly to another citrus tree, and those include disturbance, and disturbance even includes insecticide sprays. So the adults are the most mobile because they can fly. We also hypothesize that there may be an effective density here. So when you have a high amount of psyllids, they may be more predisposed to move because of high densities in the regions in which they're feeding. The last reason that I'm proposing for why they move is food quality. So if you have a tree here that has no flush and a tree here that has flush, they are going to be interested in feeding on the flush if it's available. So nymphs don't fly. That's fairly obvious from how they are on the plant. They almost look like scales. They're also not very quick to move. They're not going to get very far. Their interest is really to stay somewhere, feed, and develop. When it comes to scouting for ACP, there's really 2 types of scouting. There's passive scouting, which is using a yellow sticky card or some other apparatus in order to capture them, and then actively scouting, which is finding the pest in the grove and doing actual counts of the pest. So when it comes to passive scouting, there's some advantages and disadvantages to this, but ideally a yellow sticky card would be used and checked in 2-week intervals for management purposes. So the advantages to this are that you can really be strategic in where you place the cards, for example, at the edges of the grove where ACP tend to hang out, and then it's constantly there over time. So if you do happen to capture ACP, you are getting a presence absence. It's ideal for lower populations. So the disadvantages to this are that you really need trained labor, or yourself need to be skilled in knowing if it's an ACP or not on the trap itself. It can be expensive depending on how many are deployed and what is the replacement rate. And additionally, it can give you false negatives. And so what that means is that the card may not be catching adult psyllids, but you may have psyllids in the grove. They're just not getting to the cards. Active scouting is where I feel like PCAs and other ag professionals who are in the field looking for this pest can really be strategic in their approach. So there's really, again, 2 types of sampling methods for ACP. And the first would be visual sampling. So what you're going to do is you're going to target finding flush points and then assess the number of eggs, nymphs, and adults that are present. And you really need a 10x hand lens for this, especially for the eggs. The eggs are quite tiny. And then there's also tap sampling for adults. Some people don't really like this method, but if you don't have a lot of flush, this is actually ideal because you're not going to find the adults visually on any sort of point. And so what you do to tap sample for adults is you can use just a standard mallet from whatever hardware store and look for a branch that has a width of at least 1 inch, and then you tap that branch while holding some sort of flat surface underneath, and the ACP will fall onto that flat surface. However, this really depends on whoever is taking data here to be really familiar with what the psyllids look like, because once they have a search image for them, it's really easy and quick to count because the psyllids themselves are very disturbed by this. So they do stay on the board for a good, sometimes you have to wipe them off because they're so disturbed and they're just sitting on top of that board. So in this, it's best to take these approaches and target where psyllids are most likely to be. So the first place that they are is on the flush. So flush is any sort of new light green growth. This is really where all the action is because this is where they're going to lay their eggs, and this is where the nymphs are going to be growing. And oftentimes you will find adults also aggregating there. So field edges. ACP are really attracted to trees on the edges, and you will find them there first before they penetrate the interior of the grove. However, when you're dealing with younger trees that are farther apart, you are going to find that the Asian citrus psyllid will penetrate that grove beyond the edge a lot faster than in a mature grove. So that gives you two strategies for thinking about field edges. In a younger grove, you're not going to see as strong of an edge effect as in an older grove. And my last point here is to really know the grove. If you continually and regularly scout the grove, you'll find that there are hot spots or certain trees in which you'll find psyllids before they distribute anywhere else in the grove. So if you really know the grove, you are going to establish those hotspots. It's a great place, for example, to put a yellow sticky card. So there are some nuances to this, like anything. This is a pretty weird pest. And so there's so many different types of flush. It varies by tree variety and tree type. And it can be quite complex, really figuring out what you're supposed to be looking for. So for example, here you see this is an older branch or leaf here, and then you have young flush points coming out. And so these are really ideal, these super tiny flush points, because this is where you're really going to find eggs, and eggs are going to help you track that population through to the adults in the field. The second nuance here in the middle picture is that this really super young flush tends to be folded. And that in that fold is where you tend to find the adults and very young nymphs. So this is why that 10x hand lens is really necessary. And then once the flush starts to expand here in picture C, all the way to your right, you'll find them more so down on the leaf nodes. So those are just kind of pro tips for if you find them, where to find more and to get that count properly. Generally speaking, there's a couple best practices here. I really tend to think that a combination of the active and the passive is ideal, especially when you have low populations. If you have known high populations, meaning that you're getting 5 to 10 nymphs on a flush point annually at some point in the season, the passive might be good to possibly early detect them, but it's not going to do as much for you as doing continually active. But in many places throughout the state, we have low populations. And so a combination of these two techniques is the ideal. So really, we're looking for a biweekly frequency here in most regions from June to December. And then in low populations, especially where you know they're low, a monthly approach to the different, going to your different scouting sites is ideal. But biweekly, where you have really high populations, it takes them about a little more than 2 weeks to cycle through a generation. So in order to evaluate either the treatments or whether they're going to be reappearing, that biweekly frequency is definitely ideal. And so like I said previously, making sure to tap sample when there is no flush. This is going to be a better technique of detecting them if you don't have any flush points. And when you do have flush, it's best to combine the visual and the tap sampling. Sometimes you'll see the adults visually on the tree, but it's also, sometimes they will only be on the interior of the tree. There's no way to really tell that. So in order to get an accurate idea of how the population is dispersed, whether it's just nymphs or you do actually have a significant adult population there building, combining the two techniques is ideal. So like I've said previously, focusing on the edges of the grove, unless you have a particularly young grove, non-bearing age, that's when you'll see the distribution more regular throughout, but they will still be on the field edges first. And like I've said also previously, knowing the grove, finding those hot spots, and sort of crafting your scouting technique and approach to account for nuances within the grove itself. So there will be areas that more consistently have populations. In some groves, it can be one particular edge. In some groves, it can be one particular tree. And the younger the trees are, the more likely they are to have stable and consistent ACP populations, just because those trees are pushing flush with much more frequency than the older trees. And then again, depending on tree type, for young lemons tend to have flush all the time, and they push a lot of flush per year, and I'll be showing you photos of that a little bit later. So I'm going to continue on. This section is specifically about management of ACP populations in the field. So general best practices, really trying to dive into the complexities of organic management, and then a conventional ACP trial that we're running right now, our results to date. So in terms of general best practices, like many other pests, one of the most important things here is going to be to rotate the chemistries. It's very important for resistant management, and it will be going forward even more important as the chemicals are increasingly regulated and limited in their use. So proper application of systemic chemistries, if those are used, this really can differ by region, with some regions having soil types where they can't use them at all. And so if you have any questions about that, feel free to email me, and I'm happy to have a discussion with you about it. It's sort of beyond the scope of this general statewide talk. So really, success of ACP control depends on spray coverage. They can be hard to make contact with, but there are approaches and adjuvants and spreaders and optimizing water volume that can help spray coverage be better. For example, keeping your water volume under 300 gallons per acre with most chemistries is going to be ideal. So let's dive into organics. This is something my lab has taken on, and we're trying to figure out what to recommend here because it's quite difficult to get good efficacy with these products. Additionally, if you have a large established population in an organic grove, it's almost impossible to get really good control of those populations. And we know that because we've tried it ourselves here, and it's not pretty, and I'll be showing you some of that data. So same best practices here as with conventional chemistries, but the issue is that we're limited in the amount of products that we're able to rotate. So in fall of 2009, in San Bernardino County, we ran a pretty extensive organic trial. And so the goal of that was to look at adjuvant and AI combinations, hoping to get more residual control with those compounds. Just to give you some background on what was going on there, it was about 108 trees per acre, air blast sprayer with a 150-gallon tank, 2.1 ground speed, and then some very specific information on the nozzles here at 150 PSI. So here are all the different treatments that we ran. So just the 415 oil alone, pyganic with 415 oil, And these are somewhat very low percentages. If I could go back and do it over again, I would do a bit higher on the oil percentages. Pyganic in a product called MP'd, entrust with the oil, pyganic entrust in oil, pyganic as a direct and oil, and then pyganic, the 415 oil, and then new film P. And so this figure I'm about to show you is very messy, and it speaks to how difficult it was to evaluate this trial. So what you're looking at here is the mean number of ACP per flush. So the first thing I want to point out is look at the axis here. So we're going up to about 150 here on this axis. And again, this is nymphs per flush, and this is an average. So this speaks to how infested this grove is. So to start, you're looking at somewhere between, it looks like about 30 in most of the trials for the pre-count. And you can see that 12 days after treatment, in some of the treatments, we're having absolute explosions with sometimes 75 or even more than 100 nymphs per flush. So it's a hard study to interpret. And one of the things that was complicated here is the way that the treatments were laid out in the field is that, like I said, the distribution tends to be more towards the edge. So we tried to take advantage of that by using mostly edge, but by the time we got to the pyganic that's on the interior, you can see the pre-count here, which will be the blue bar, has nothing to start. So this is just already getting deep enough into the grove that we're having less good populations to start with. And so this is a messy figure, but I'm going to give you some really broad conclusions and give you an idea of what we're focusing on this year. So ultimately from that, we kind of determined that, you know, mixtures of pyganic and entrust as a direct and new film may be more effective, especially than entrust alone, while pyganic was really more the star product there. 12 days in a huge population like this appears to be the maximum duration of control. And so we really needed to redesign the study for a smaller, looking at this at a smaller scale with a more reasonable population. And so this fall, you know, this is going to be going out in the field in a couple weeks. We're going to be looking at the mixture specifically with pyganic, because pyganic seems to be the standout product in terms of efficacy, and increasing the residual of pyganic is going to be incredibly important going forward. So that we, you know, especially when we're doing area-wide treatments and organic growers are participating, we can really help optimize what's going on. And furthermore, mixtures of pyganic and entrust, that's a very expensive thing to ask people to do. So really optimizing at the pyganic level is the goal for this year. Okay, so let's keep it moving. So conventional products, this year we are almost at the end of running this trial. So these are sort of newer to the market products, especially Safina was approved for use in 2020 in citrus and throughout the state. And then Savanto is a recent product by Bayer. So the trial here that we're running right now is a max label rate on Safina, Savanto, and Movento. There was no adjuvant used in this trial, and that's because I didn't want to make the choice of one of them because I'm not sure specifically how the different oils are going to interact with the AI, so we kept it clean and just used the AI. So this particular trial is about 100 trees per acre. It's in Palma Valley, which is in San Diego County. These are younger trees. So it's a speed sprayer. And here's more details that I won't pretend that I didn't understand. Irene Davidson is the one who applied this. We're at 100 PSI and 200 gallons per acre. And so one of the exciting things here is that Movento is a great product and used widely and people love it. But Safina and Savanto are substantially cheaper. So that is another angle to the approach here. And so I'm going to get to show you a much cleaner figure than the one for the organics. So here's what we're looking at here. So again, the blue bar is going to be our pre-count. That is before we applied anything. Red bar is 1 day after treatment, 3 days after treatment, 7 days after treatment, 14 days after treatment, and 21 days after treatment. We have another one that is going to be 28 days after treatment. So we're looking at a full month here. And so this red bar signifies, this red bar that's going across the graph signifies the proposed limit mean number or threshold for psyllid. So it's 0.5 nymphs per flush. And as you can see, the untreated control stays above that for the entire part of the trial. Our Savanto results are quite weird. I don't know if they were moving at a certain point, or that we didn't get good control on eggs here. There were mostly eggs in this count. Then by the time we came out in the next one, we had young nymphs. But after 7 days, we're keeping the mean there underneath the threshold. So it takes that first 7 days to get any sort of efficacy with that product. Movento has a beautiful curve here where at 3 days after treatment, we are significantly below the threshold there and continue to be throughout. Savina takes a bit longer to fall beneath the threshold. So even though this looks like it's below the threshold, our error bars there are saying that that looks a lot different. In practice, you could have up to, 1.5 as your mean number of nymphs per flush. And then as we go out to 14 days, we're not finding anything. And then we're keeping that quite low there at 21 days. Another thing that may have impacted this trial are the average temperatures in Palma Valley. So from the pre-count to the 3 days after treatment, our average high per day was 91 there with a low of average low of 60. And the highest temperature that it got within that week was 96 degrees Fahrenheit. On 7 to 14 days after treatment, we're looking at 102 max. So that is quite hot. It's yet to be determined really how hot it needs to be to kill off the nymphs. And then at 21 days, we've got a 104 max. But on most of the days, and that's why our averages are coming out like this, it wasn't extreme heat. So it's great because this really is a look at a range of temperatures that really is experienced throughout California, maybe less so on the coast. But it goes to show that, you still are going to have a low level of nymphs there, but it is well below the threshold with those treatments. So in terms of conclusions, again, these are sort of preliminary results. We also have data for adults, and we have one more date to include here. ACP and field trials, it's pretty difficult to work with because their distribution can be spotty throughout the field. So we have to do a lot of pre-scouting and then kind of select populations that are more similar in their density. We definitely found that Movento had the best results, but Safina was a strong second. Both have translaminar activity and take a little bit to work. So not finding very high control 1 day after treatment, but once you're out at 7 days after treatment, seeing nice results. So the translaminar activity we're thinking is likely the cause of that delay. Savanto, we had a bit of an odd result here with the nymphs. This product also has translaminar activity, but a little bit less so. We do think the temperature likely has helped suppress and control the population in that study. And so one thing that is for sure is that the control with these products is likely going to be greatly improved with adjuvants, in particular, adding in 1% or 2% of 440 oil. And so another thing I'd like to point out is that Movento is a really great product, but it's, let's not overuse it. We want to keep that around so that we can rotate, in particular, save that for a more broad spectrum control, especially if you've got a scale outbreak or a mealybug outbreak, which seem to be common things that are happening. So here I'm going to be talking about new technology for ACP management. Just two different things that my lab's looking at currently. So red dyed kaolin and essential oil repellent bars. So here's our red, or it looks pink in the field really, kaolin. Here's an aerial photo of our trial that we're running in Ventura County. So there's really two hypotheses with this study. One is that the red dyed also will enhance plant growth more than the white. We haven't collected our second set of data yet on that, so we don't have data to present on that yet. And the second hypothesis would be that the red kaolin repels ACP better than the white. And the theory there is that the visual cue for ACP is more disruptive when red because it's blending more into their visual. And so here is our field results from a Riverside grove. It's an older grove that is bearing. And I would say that the grove is maybe not in the best condition, and so flush tends to be quite inconsistent. And then let's look a little bit at this chart here. So we're looking at average number of adults that are coming off in a tap sample, and we have a lot of time here. And it's really important to point out because this treatment does wear out, the plants do grow out of it. And so the treatments were applied June 9th of 2020 and then August 15th of 2020. And that's what these black bars are signifying, so that we're looking at them post-application. And this older grove, in terms of tap sampling, these are pretty inconclusive results. These trees are huge. They're hard to get good coverage on. We don't think that older trees are going to be the best way to use this. And that's definitely coming out here in our results. So adults are not as important as nymphs, but this is the cleaner, ready to go data set that we had so that I could present something on this. As you can see in this study, it does look like the white kaolin is doing better than the pink, which is contrary to our hypothesis. Now let's go to a younger grove. So this is Orchard Farm, managed by Limonera and Associates Insectary in Ventura County. And similar to the last graph, we're looking at tap samples of adults here. And so in this one, our application dates, we've had 3 applications. This one on October 1st, there's no data post-application for that one yet, but May 21st, and then July 15th. So in this, with the younger trees, we are finding that the pink is doing better more consistently over time. All these different colors represent different dates. I know it's hard to look at. That's why I think the best way to interpret this chart, especially because there's very low population here, because it is very highly managed young grove, you're just looking for how many you are starting to see. And why would there be more in the white? Well, we think that has a lot to do with the theory behind the red kaolin, in that the flush points themselves, even on the white, are even more obvious than on the pink. And so we have photos here of when they're super grown out. And so while this might look more visually to us like, wow, that flush is really sticking out with the red kaolin, for insects, that red is actually more blending in than the white. The white is more likely to be making the psyllids, you know, it's like a backdrop or a stage, you know, presenting the flush to them. So that's what we think right now in the short term based on our results. And one of the biggest complications that we're seeing with this as a treatment option is that they're really growing out of this quickly, and it's needing to be reapplied every 4 to 6 weeks, which is pretty complicated when you're trying to fit it in with other applications that need to be applied onto these plants. So getting this removed from the fruit, working with packing houses, what does the marketability look like long-term? That's an issue and a concern. However, you know, on non-bearing trees, young trees that are going to be more attractive to psyllids, this could be a very strong option, but again, needs to be applied every 4 to 6 weeks. We may see a better residual with the red kaolin, but that's yet to be determined from the data. In Riverside, on these older trees that are not doing, you know, not in the best health, not maintained in the best way, you know, we're seeing 10 to 12 weeks of coverage, and we're hoping to extrapolate more from that with our nymph data. And so that'll be something that will be either published on or presented on later this year in early to mid 2021. So another issue that we're running into with the red is that currently that dye that's in there is not organic approved. We tried to run it through CCOF and it just did not, you know, there's 1% of some proprietary ingredient that they won't give up. So that's still an investigation of between me and my colleague, Christopher Benson at University of Florida. So another thing my lab's investigating are these essential oil repellent bars. This is one hanging on the tree in the picture here. So they're 20% fir oil. The field trials will be done on young trees, and that's really because we're trying to figure out density, and we don't think it's going to be a practical tool for a large full-grown tree. The densities we're investigating are blocks of 25 trees, and we'll be distributing the bars on just the edge or every tree or every other tree within that 25 tree block. We're assessing repellency over time. One of the great things about this is it is organic ready. They're fairly cheap. I think they're about $1 per bar, but there's no commercial pricing on it yet. This trial is going to be placed in coming weeks. So I'm going to try to speed through this last section here. I just wanted to give everybody an awareness of a new project that's up and coming. I had a lot of good response when I presented on this, so I figured I would share it with you guys as well. Beth had a statewide scouting project, and this is the continuation of this. We have different questions. So really the first thing we're trying to address with this new version of the scouting project is what percentage of HLB positive ACP are in commercial groves. That's a key question that I think growers need an answer to in order to assess risk properly. And understanding the phenology, which is a fancy word for the presence over time of HLB in ACP. So do we find that they're more prevalent in the spring or the fall, et cetera? Trying to find an answer to that question. And thirdly, which people do tend to be fascinated with, is that what percentage of ACP color morphs is in the field. And this has downstream implications, which I'll go over in a second. This figure, it presents what CDFA is currently found in terms of HLB positive ACP in the quarantine zones. So highly distributed throughout LA County and then of course in Riverside, but surprisingly they don't tend to find them as often as you would think. So this is going to be quite interesting. Again, this is backyard work. They're really not looking in commercial. So we'll be addressing that through this grant. And the methods here is that we're going to really we're going to focus on sentinel sites. So we're going to look at Beth's data from her previous scouting project and really pick out in Riverside, San Bernardino, Ventura, and San Diego County, 15 sentinel sites that we're going to look at over time. So we're going to be doing monthly systematic sampling of whole insect community using a DVAC, which is basically a large vacuum. And this is going to be focused on grove edges. If there is flush and nymphs available, those will also be included. There will be a lot of processing in the lab, so all of those psyllids will need to be sorted by abdomen color and processed and set out for HLB testing at the University of Arizona by Dr. Judy Brown. So this is going to amount to about 13,680 samples processed for HLB over 2 years. So let's talk about the morphotypes. People think this is exciting every time I present on it. I'm so excited about that because I think this is fascinating, and I'm excited to see what this looks like in California. They do have numbers on it for Florida. So what I mean by morphotypes is just these 3 different colors on the abdomen that you find in populations of Asian citrus psyllid. So this first one all the way to the left we call blue-green, the center one we call white-gray, and then all the way to your right is ones that we call orange. And so those abdomen colors do have some effects in the sense that, well, let's talk about where the color is coming from. So this is really nerdy entomology stuff, but basically insects don't have a liver the same way that we have it, in that it's a certain organ. It's actually this weird substance that kind of floats throughout their open skeletal body. And it's called fat body, and there's pigmented cells in that fat body, and that's what's creating the colors that you're seeing on the abdomen. And so the blue-green ones, they're really, in the literature, the most long-distance flyers. They acquire less of the bacterium that causes HLB than the gray or the orange morphs. So they are thought to be less inoculative, meaning that they're less of the spreaders of HLB. The females tend to be heavier than in the other color morphs. And then the gray and orange morphs, what we find is that they have a higher symbiont load, and so that they carry more of the bacterium that causes HLB, which here is abbreviated as CLAS. Furthermore, the orange ones are more susceptible to insecticide. They tend to have less detax activity in their internals. And it's important to know if these vary in proportion throughout the year because it may have some implications for some of the insecticide control. So we're not focusing on nymphs here because they're really cyclically available. If there's flush, you might have nymphs, but the adults are able to be present more thoroughly throughout the year. We're also going to be separating out teneroles, which is another fancy science word that is just, all it really means is that once the nymph is going from their pupal stage to the adult, they have this almost albino look to them, as if they've just hatched and their wings are kind of drying, sort of like what you would think of with a butterfly. And they tend to have, at least in the literature, higher loads of CLAS. So we're going to be separating them out separately as well. The DVAC sampling really eliminates the sampling bias of scouts and the seasonal variation and access to adults. So scouts can be more or less better trained in finding psyllids, better at tap sampling skills and knowing when they've done it well. And so the DVAC for my staff will eliminate any sort of bias between who is sampling. And also when adults are kind of hiding, either in the winter, deeper in the tree, the vacuum will allow us to pull those directly out. So the deliverables from this project that we're hoping to give here is that we're trying to give up-to-date data for growers specifically on the vector population in their region. Try to figure out what the percentage of HLB positive Asian citrus psyllids are, which is really key in risk assessment. This is how the disease is getting around other than humans moving psyllids and grafting plants that are diseased. And then also we'll be continuing to evaluate the efficacy of ACP control programs in conventional and organic citrus. So just like Beth was doing, we'll be pulling the pesticide use reports and trying to assess what the treatments tend to be doing. So I'm at the end of my talk here and I have plenty of time left over for questions, but I just have a couple of thank yous, especially my lab who has been successfully working during the pandemic. None of this is possible without them. Alan Washburn and Irene Davidson, who I've worked closely with to get the Kaolin and the ACP trial and another ant trial all applied. So we've really been hanging out a lot. Brett Chandler from Associates in Secretary, who's been applying the Kaolin trial in Ventura County. The Citrus Research Board for their funding and other support. USDA MAC, which is a funding program that funds the repellent bar project, and all participating growers where I'm using their land, so Limonera and then Washburn and Sons. And with that, I can take any questions.
Okay, and we have quite a few questions, so hopefully we can get to them all. The first one is, it has been proposed that the CPD-PD program increases the trapping interval in the San Joaquin Valley from 2 weeks to 4 weeks between servicings. Do you believe this is a good idea?
I do not believe that's a good idea. Right now, while that region is under eradication, I think it's best to stick with 2-week intervals. I understand it's a lot of labor, but I really believe that that 2-week interval is important for psyllid development and detection when you're using a tool that's not great the way that a yellow sticky card is.
All right, and then we have another that says, in many Southern California groves, there are wide picking drives dividing blocks into smaller segments. Would you include these edges when scouting or placing traps for ACP?
Yes, anytime that you have any sort of divide, you're looking at an edge. Even if you have an uneven border, that's creating even more of an edge. And so when it comes to scouting, looking at all of those to start, and I promise you that when you start to get high psyllid populations, and that if you continue doing this over time, you really will find that there are hotspots that you can target and really start to hone in on your grove and figure out where they're going to be first. And this will help greatly, I think, with management and keeping populations below the threshold.
Thank you. And then we have a question from John asking, is there a temperature effect on tap sampling?
So that's a great question. So what we find is that temperatures in the extremes, so if you're going first thing in the morning, and for example, in Palma Valley, those temperatures when we were going out there, even during peak heat, you know, you could be out there and it's only 58 degrees. So going early in the morning when you have lower temperatures, and then if it's really hot, they're also going to be pretty arrested. However, we do think that when it's really hot, they do move more towards the interior of the tree, and it might make the tap sample less effective.
Thank you. We have a question from David asking, what is new film?
New film is a spreading agent, and that's all I actually know about it. It's just a product that was recommended to me actually by San Joaquin Valley growers when we were developing that organic trial, something that they think is really effective in getting that material, the materials to spread and stick to the plant.
All right. We have a question from Curtis asking, has insecticidal soap been tried for an organic solution?
Not that I know of. Every time we've used insecticidal soap in the lab to clean up plants, it really kind of isn't something you can just let rest there. When we clean plants in the lab anyway, there tends to be a little bit of a rubbing action to get whatever is off of there. It's also unclear how that would bind and how temperature would affect it. So I don't know whether it's been tried. I haven't heard anything about it. That's sort of my best answer and best guess at how that might go.
Okay. And then what is the devastating contaminating impact to adjacent growers of an organic grower? Not quite sure.
So if that organic grower isn't properly managing and keeping their psyllid populations between the threshold, you're going to get reinfested from their grove if they have a high density of psyllids. That's just the reality. There's a couple of other cultural tools you can use, but it takes a bit more of an investment. For example, screen fencing or putting up some type of natural barrier like a tree line, a windbreak, that type of thing. So it does take more investment on your part, but at the same time, I think it's a worthwhile investment if you have a grower that's either, well, in particular, a grower that refuses to treat. That is the biggest issue I've seen. Managing psyllids with organic materials is possible, but it requires diligence and thoughtfulness.
And then we have a question from another attendee asking, are the three products Movento, Savanto, and Safina in different categories for resistance management rotation?
I think so. Yes, they have different modes of action. They're in different groups, so they can be rotated, the three of them.
Okay, so there's a question from Devin that asks, do hot temperatures reduce the ACP population?
Yes, and so we're trying to get a data set on that as soon as we can. We're doing sort of some side projects where we're caging psyllids out in the field. And for example, when we had really high temperatures in Riverside, over 100, we had placed, so what we would do is we would take 10 males and 10 females and let them lay eggs on a branch with flush, and then it was caged out there, and then we would go remove the adults and count the eggs that they laid. So we had one sleeve out there where we had over 200 eggs laid, and after experiencing those high temperatures, we ended up with 2 nymphs. So those high temperatures absolutely influence the ACP numbers, and you can definitely see that when you look at the desert numbers. They're have a much easier time keeping a handle on their ACP populations.
Great, thank you so much. There's a question from Margie. Doesn't kaolin suppress beneficial insects?
That's a difficult question to answer, Margie, because I think it really depends, right? You're not going to have any insects there. You're going to have a less of an insect population there for the beneficial insects to be feeding on and to also be engaging with. So the question is it harming the beneficial population, or is it causing them to move to find other resources and not be as established in the grove? However, in those younger groves, I kind of doubt that there's as strong of a beneficial population than in an established grove, because there's just simply less refugia. So that's my best job at answering that question for you, Margie.
We have one from Brett that says, which formulation of Pyganic was used?
So the formulation that is in the field trial that we ran last year was the 1.4. And so that was a big mistake on our part. We should have used the 5.0. We're using the 5.0 this year. So just to clarify on that, that's a great question.
Okay. And then Robert asked, isn't Movento a systemic?
So systemic is the way that I look at this, and this may be wrong, and feel free to tell me I'm wrong. If you have a foliar application, you're going to be getting more of a translaminar activity than with a drench, where the product is being resorbed into the entire plant. That's what I consider systemic. That's my best answer to that question.
Thank you. And then Emily asked, the new conventional pesticides, are they neonics or are they better for bees? How do they compare to Actara?
So Actara, as I know, is a neonic. And so none of these products are in that specific MOA class. And I really think that the bee thing is a little bit too complicated to comment on. As soon as something is called bee safe, bee whatever, you're gonna have certain scientists that don't do field work claiming that in the lab you're having toxicity on bees. So that's really my best answer. I'm not claiming that any of these products are particularly good for bees, but in citrus, when you're applying them outside of bloom and distance from bloom, you should not have any interaction with the bees, because the bees will have no interest in the citrus trees because there are no flowers.
Excellent. Okay. I do have a question from, let's see, I'm trying to prioritize because we still have quite a few questions. Derek asked, is Safina on the UC ACP material list for area-wide management and or the spray and move list?
It's not on the spray and move list, but I think it is on the area-wide management list.
Okay. And then, are you going to be spending more time in VC, the heart of the lemon growing areas? We need to modify the timing of regional spray areas. The advisory committee has lots of new information since Beth finished up.
Yes, I mean, I would love to spend more time out there. The thing is, I think that the biggest challenge in VC is just getting the equipment to, and VC is Ventura County for those people who aren't from there and don't recognize that. So their biggest challenge, to my understanding, is getting equipment and getting it dispersed across the county. So how we resolve that issue is going to be more complicated than what chemicals and timing.
Thank you, Monique. We do have a couple questions about fir. One person asked, what does FIR stand for? And then another person asked, the fir bars, what is the bar made of?
So the fir oil, this is just an oil. I mean, it smells like pine trees. And so let me think about, so what was the second part of the question? Oh, what is it made of? So it's actually in glycerin. They're not much different than soap on a rope. That's kind of our research joke about it, is that these are scented soaps on a rope, and they do evaporate over time and release the essential oils. Again, we're just starting to get into playing with them. We did do some work on how quickly they evaporate in California, and it looks like, you know, they might need to be a month to 6 weeks, and again, more ideally on small trees. But yes, they do fir oil, just in the sense that it smells like fir trees. It doesn't stand for anything.
Okay. And then one person asked, one of the insect monitoring supply companies markets a lure for ACP. Have you used it, and is it effective?
I have never worked with an effective lure. I know which one you're talking about. I'm assuming it's an AlphaSense lure. May you capture numerically more psyllids on that trap with the lure versus one without? Somewhat likely, but not numerically. You know, when you look at the statistics, when I've worked with that data before, it's not significantly more, but numerically more it is. I don't think there's much of a difference with the sort of early detection principles there. You know, the one with the lure is not necessarily more likely to catch the first psyllid. It's really a very short range thing for psyllids, and they are more tuned to visual cues than olfactory cues, meaning scent.
Okay. Jamie asked, are you planning on doing any studies using imidacloprid?
I am not planning on doing any studies on imidacloprid. In fact, in combination with Dr. Frank Byrne, we have a couple projects that we're going to be working on looking at neonic alternatives. And so some of the products presented here are proposed alternatives. As you may be familiar, the state at the DPR level is in the process of limiting the way that the neonics are used. And so we're trying to stay on top of that to give you best practices for using alternatives. So no, I don't have any planned work for imidacloprid or any of the neonics.
Jose asked, are you evaluating the load of the bacteria on the ACPs or in the trees sampled?
It's only in the psyllids, actually. We're not doing anything with the trees. Part of the reason for this is regulatory, in the sense that if you are currently under the regulations, as is my understanding, Victoria Hornbaker is on the call, I think. I saw her answering something. She may or may not still be here. If you have a positive plant sample, I do think that the current regulations state that you still need to remove the plant. And so that is part of the reason that we're focusing only on the psyllids, but the primary reason is really that they're the mobile things. They're really the spreaders of the pathogen. And so understanding what percentage of them is carrying the pathogen is really key to risk assessment.
Okay. Is there some way you could release the data in some form from your sampling project on a regular basis, say quarterly? That's from Sylvie.
So what we're hoping to do and what's actually written into that project is to do kind of precisely that, but it would be more in a meeting form rather than an email or a document. We're hoping to host some regional webinars again, and of course the San Joaquin Valley people will be invited to attend whatever one they find most interesting. Especially if we're still remote, then everyone will be able to join in a webinar form like this. However, if we're in person, then we'll be traveling around the state to meet with each region.
So one person asked, have semiochemicals been identified or tested for use in monitoring or management programs?
Yes, extensively. I did a lot of that work when I was in Florida. I just think it has to do with the sort of rank order of how psyllids are perceiving the world. And so their primary is visual. And so if you focus on an olfactory, you're never, and a thing that's really important to point out to people is that these are short range cues, right? So the reason why those yellow sticky cards, with or without the olfactory, they may simply just not be more attractive than the flush. If you have that semiochemical attractant, so to speak, it's never going to put out the same amount of volatiles or maybe the volatiles that the insects themselves are most accustomed to compared to the flush. So that's why it's not a great detection tool. However, at short range, and when there's no flush especially, those cards are going to be attractive to psyllids that are present.
Great. Devin asked, acephate or orthene is not currently labeled for psyllids, but is there any interest in trials for young or non-bearing conventional citrus?
No, and I haven't thought about or considered either one of those products thus far.
Okay, Pam asked, how effective is ant control at reducing ACP populations?
Well, the whole idea of ant control is to allow the biocontrol agents to help reduce the population. So if you don't have established biological control agents, then your ant control, the ants, in my understanding and experience in the field, are more likely to go after aphids and mealybugs than psyllids. And so when you think about the honeydew that those three insects are putting off, your mealybugs and your aphids are putting it out in liquid form, where the psyllids are putting it out in that string, and that's a waxy cover. So that's less preferable to them. They will still tend them. However, in the lab, we're investigating whether or not ants can kind of do what we're calling code switching, where sometimes it makes sense to tend them, sometimes it makes sense to eat them. And that's going to really depend on what else is in the field. Do they have a protein source for Argentine ants as well as another sugar source? these are questions we're trying to dig into, but for Psyllid, again, you're controlling the ants for biocontrol agents, not necessarily because they are going to make your treatments less efficacious.
Great. John asked, do the majority of adults fly horizontally as indicated by edge effect or by wind? If horizontally, then a barrier planting or screen would be, then would a barrier planting or screen be effective?
Yes, the barrier planting or screen would be really effective. And I was really skeptical about this until we visited a grove in Florida. So we used to go out with a big DVAC and collect thousands and thousands of psyllids for resistance management tests. And the most psyllids we had ever collected was at this one grove. And the part of the grove had this tree line around it. And when we went inside that tree line, there was just enough space to move a tractor in and out of it. The psyllid populations were exceptionally low. You can almost not find them at all. And so I'm a big believer in that tree line and barrier strategy. It's just a lot of money and input and potential water use to strongly suggest. So it has to be really optimized to a particular grove situation. For example, interfacing with a mismanaged organic grove or residential.
Okay. Robert asked, do you think that high temperatures with low humidity would be more effective than high temperatures with higher humidity?
So the humidity is going to limit how high the temperatures can get. Here in California, we're mostly dry, but once you hit a certain threshold of high temperatures, you're not going to sustain the same high humidity. So in Florida, it gets super hot there, and it might feel like it's 105, but really it's only 98, and that's because the humidity is sort of insulating the temperature from going that much higher. Here we're really, we tend to really experience, especially more inland, super high temperatures that are also very dry. I think it's really the temperature itself and really getting up that high and being dry. I do think that does to some degree play a role, but more so in the amount that the temperature is able to go very high, rather than the humidity being something that is, you know, helping those high temperatures, if that makes sense.
Okay, Chad asked, do we know why some color morphs act as better vectors?
We don't know specifically. However, I have a hypothesis that I'll share with you. So for those orange morphs and the white morphs, one of the things I mentioned is that they have a higher symbiont load. And if you've been following the work of them trying to culture the bacterium, the only way that they've been able to culture the bacterium is with other bacteria. So clearly the HLB sea loss bacterium is dependent on something else in those bacterial communities to survive and thrive. So the more of those symbionts are, you know, my hypothesis would be that the symbiont load would help support the sea loss growth and replication within the insect itself. However, that's not tested. That's just an off-the-cuff hypothesis for why.
Okay. Greg asked, is there any work being done to correlate or refute the effect of ACP control on California red scale?
No, and I think that's a hard question. I think some of the there's very strong evidence for a lot of the outbreaks of the pests happening, you know, may be because of the increased spraying for Asian citrus psyllid that's widespread. So it hasn't been investigated, but it's certainly been hypothesized, and especially pyrethroids wiping out all the biological control agents. I would tend to stay away from pyrethroids as much as possible.
Okay, I'm just checking the remaining questions. Kathleen asked regarding the red dye for the, I think it's regarding the red dye for Kaolin Clay. Is there any way to make it to know what they use for the dye besides red, like more specifically, I guess?
So it's iron oxide. That's what's, you know, we're looking at less than 1% of a proprietary sticking agent that binds the red to whatever it's being applied to, and whatever it's being applied to would be an organic source, so either kaolin clay here in this case, or for mulch, wood, or in more technical terms, lignin. Again, like I said, it's proprietary. I don't have a lot more input further than that because they're not revealing what that less than 1% of this ingredient is. However, I can assure you that we're hard at work trying to figure out how we can make this an organic option, and that includes and is not limited to looking at dried beet powder and iron oxide itself and seeing if we can get it to bind to the kaolin enough to get efficacious treatment.
Okay. Jennifer asked, since kaolin clay is a type of soil, wouldn't it make a slightly impermeable surface layer when sitting on top of the soil?
So I don't think kaolin's really a type of soil. It's just a clay. I don't think, again, that's sort of beyond the scope of that study. And it really also depends on how it's applied. Yes, it tends to get on the ground, but investigating it further, I'm really not sure. We're going to have to wait, watch, and see what happens there with that soil, especially after this winter. So what the pictures you're looking at are applications that are post-winter. There hasn't been a lot of rain to wash or run that down. So, you know, we'll see.
Okay. I'm not sure what this question is referring to, but it asks, what about insect control? I thought there was a new discovery that was very promising. Is it being used anywhere?
Maybe they're talking about the new peptide. So rather than insect control, looking at peptide control for the bacterium itself. I think that the release of information that this is a cure for HLB was quite preemptive and you know, pretty irresponsible for them to say that. That compound has not been field proofed, so it's a lab idea. It's been lab tested. It also has not been formulated for use. It's unclear whether growers and others will be willing to say and do trunk injections with that compound. How long does the compound last? How frequently does it need to be applied? And does it work on HLB that is not expressing itself in its full-blown symptoms? So that's my best guess at what the question was and my answer to what my best guess on the question was.
Thanks, Monique. There's another one from John asks, I'm not sure if it's a question. It says, leading to the time of day sampling recommendation. Maybe they're asking about what time of day you should sample for ACP.
I think the earlier the better, because they're going to be a lot more arrested when they're exposed to the cooler temperatures. And you're also going to see, you're not going to have that effect of it being so hot that they're moving to the interior of the tree. So my team also aims to get out there as soon as possible. And I would recommend that to anyone else. But if you have a full day of scouting rows, well, you do what you can. The nymphs will always be there if they're there.
And those are the last of the questions. So thank you everybody who stayed on. Thank you so much, Monique, for presenting and for answering so many questions. And thank you, Joey, for being here. And thank you to everyone for attending.
Thank you guys so much.
Have a great day.
Definitely.